Hello everyone, I'm presenting this work towards the calculation of protein stability in wild type and mutant proteins associated to Rett syndrome. To start, Rett syndrome is a neurodevelopmental disorder that is mostly present in girls, is characterized by the deceleration of head growth, autonomic dysfunction, loss of language, and motor skills. Patients with this condition also have the presence of characteristics from the autism spectrum disorder, and for Rett syndrome, it has been associated in 90% of patients to mutation in this gene, determining that the function of this protein encoded by the same gene is critical for the normal neurodevelopment. MECP2 protein is a pleiotropic protein that belongs to a methyl CpG binding domain, MPD family, and is widely expressed among various tissues with the highest expression in the brain. This protein represents an important epigenetic modulator of the transcription, controlling chromatin architecture and gene expression through methylated DNA binding. Additionally, it has been indicated that this protein acts as a transcriptional activator in brain subregions, suggesting the significance of this protein. There have been reported more than 900 MECP2 mutations today. Different types of mutation in this gene have been identified, which include missense, nonsense, insertions, and deletions. Efforts has been displayed in order to apply pharmacological therapeutics for targets downstream of MECP2. Despite the efforts performed, it is still necessary to develop novel methods to gain insights into the molecular mechanisms relying on MECP2 functionality, with the future perspective of acquiring possible therapies to treat Rett and help clinicians in early diagnosis. In this context, the present research project aims at simulated by MD a select subvariant of MECP2 associated to Rett syndrome. This includes simulated the intrinsic folding stability of a set of relevant missense variants for this disease and also to determine the implication of the amino acid change in question of this phenomena. In this research, through the use of MD simulations, we could determine the intrinsic folding stability of missense mutant proteins. Simulations were performed on the software GRAMACS, considered conditions that were tested previously, which were selected for the ability to see unfolding events during the simulation procedure. Results analysis were performed through the examination of different metrics like TM score, RMSF, and RMSD2D. Previous work made by our research group demonstrated how protein conformational changes can be estimated based on MD trajectories. This model was obtained through the correlation of the unfolding fraction with the size of protein variants and simulation time, resulting in an empirical model that aims to help to a calculation of mutant protein stability. MECP2 mutants selected were subject to a protein conformational analysis, a proposed model for calculating the stability of mutant proteins and thus corroborating the reliability of it. These are one of the results that were studied in this research, and also we can consider that the results obtained by this calculation and the prediction of the protein stability were classified in three categories: tolerant, non-tolerant, and possible non-tolerant. These findings coincide with previous studies, which indicate what effect of the mutants will have. In this case, mutants result in a classification of tolerant, tolerant, and non-tolerant respectively. These findings demonstrate the reliability of applied molecular dynamics methods in obtaining correlation for genotype and phenotype. We can also mention some characteristics and some drawbacks of this research. In this case, we also need to understand which are the limitations of this study and which can be improved for future methodologies.